Hello, and welcome to Module 2, Segment 6 of the biology portion. Today we are going to be discussing and giving a basic introduction to the nervous system. Now, this is obviously part of a much larger topic, the nervous system itself. A lot of what you're covering here today will become the fundamentals for segments exploring the endocrine system, exploring further into the musculoskeletal system, and indeed for a lot of the training segments we'll be doing later on. So there'll be a significant amount of material that will not be covered today that will come up in later segments. So what is the nervous system? Well, the nervous system is comprised of, honestly, more parts than even listed here. The old traditional notion of the central nervous system being the brain and spinal cord and the peripheral nervous system being everything else is actually being called into question. And we'll talk a little bit more about that later. Now, the central nervous system is essentially, as it states here, considered the brain and spinal cord. These are, the brain itself is encased in the skull and the spinal cord is encased in the vertebrae. And typically speaking, anything outside of that actually gets transmissions via root ganglia is considered part of the peripheral nervous system. However, there is some disagreement with categorization, and certain sensory nerves, particularly the retinal, the retina, the optic nerve, and olfactory sensors, are actually considered part of the central nervous system as well. That is due in part to the fact that the nerves, the axons, are relatively short and insert directly into the brain itself. But of course, that will be a discussion for a later point. The peripheral nervous system effectively represents everything else, and that is the nerves and ganglia that are outside the CNS. Now, a lot of these structures, a lot of these segments that are considered peripheral nervous system actually insert into and actually lie within various parts of the central nervous system. So it's important to keep in mind that these are not clear-cut definitions. And when talking about CNS versus PNS, bear in mind that there is still some overlap. So, what are the types of nerve cells that comprise the central nervous system, peripheral nervous system? Well, there are a huge amount. And in fact, one of the most fascinating things is that we still, to this point, are nowhere near categorizing every type of nerve cell that is located in the brain. There could be hundreds and hundreds, if not thousands of types of neurons within the brain that we still have not actually managed to categorize. However, we can basically differentiate the two types of cells in the nervous system as the neurons and the glial cells. Now, neurons, if you think about it, are basically the communication cells. These are cells that rely on their ability to generate a charge or maintain a resting electric potential and transmit that potential to other neurons. As we'll see in the next slide, every neuron basically consists of the soma, which, if you think about a normal cell, if you look at the neuron, the soma is the part that kind of looks like a regular cell. It has has the nucleus in the middle of it, has all the organelles and everything else. That is the soma, the body of the cell. Now, a neuron also has an axon. And you'll see this, of course, much more notably in the neurons of the peripheral nervous system. The axon is the long segment where the action potential, or think of it as the output arm. At the end of this axon is the axon terminal. This is where the signal propagates. If you think about really long neurons in the human body, like let's say the neuron that travels to your big toe, which is extraordinarily long, that is almost entirely comprised of one long axon. Now, the dendrites are actually the input arm. If you think about the neuron, and again, we'll see this on the next slide, the soma itself is surrounded by all these little branches that come in. These dendrites are kind of input arms. They're places where other axons can actually attach to transmit signals to the neuron. The second type of cell located in the nervous system is very different. Whereas neurons are all characterized by their ability to transmit these action potentials, the glial cells comprise many different kinds of cells. And these are all the support cells that are located within the nervous system. Now, they play many roles. They, for example, here listed, create and secrete CSF, or cerebrospinal fluid. That fluid that's actually located in your spine itself and serves a variety of purposes, ranging from aiding in oxygenation and aiding in fighting off infection, etc. That cerebrospinal fluid is actually secreted by special support cells in the area. The myelin sheaths, which are located around axons, which we'll take a look at in a second, there are separate cells that actually generate those myelin sheaths and actually comprise the myelin sheaths itself, secrete that protein and form into those shapes. Immune protective response. The nervous system, of course, has its own sort of immune system, its own sort of a little ecology in there. Everything from your brain to your spinal column, everything else. There are special glial cells that are responsible for aiding in inflammatory response, anti-inflammatory response, et cetera, et cetera. Now, neurons themselves, and in fact inside your brain, it's very important that these clusters of neurons, which aren't always perfectly vascularized, have their own way of circulating blood, circulating oxygen, etc. There are glial cells whose entire role is creating sort of this mesh network of oxygenation, etc., that helps keep these neurons alive and support them. So basically think about it that way. Anytime you see glia, glial cells, or anything else, just think support, and neurons are communication. Here is a basic neuron. As you can see, the soma is on the left with the nucleus in the middle of it. The dendrites, those inputs, come in around it. Now, the Schwann cells, as you can see, are actually the glial cells that are responsible for generating the myelin sheaths. And you can see that those myelin sheaths are wrapped around the axon. We'll explain why those exist in a moment. Some of you may be familiar with it, but it's obviously worthy of review. Another important thing to look at here is the node of Ranvier. Now, those are located in between every single Schwann cell and represent these sort of, I don't want to say dead zones, let's say exposed zones in the axon between every single Schwann cell. So those are important parts to take in right now, because what we're going to talk about is the difference between myelinated neurons and non-myelinated neurons. All right, but okay, so this is what a neuron looks like. So how does it actually transmit a signal? Well, if you recall the potassium-sodium pump, now we were looking at, when we discussed cell membranes, we were looking at channels, we were looking at various gated, we were looking at carrier proteins, channel proteins, gated proteins, etc. And then a little bit in the last segment, we talked about how ATP itself is responsible for opening and controlling some of these gates. What a neuron effectively does is it maintains a gradient where sodium is continuously pumped out of the cell and potassium is consistently pumped in. Now, the action potential, the neuron itself, is always trying to maintain a negative charge, and we'll talk about why that's important in a moment. Now, opening these gates, when these channels are either opened or closed, because what's happening is the neuron is constantly pumping sodium outside of it. Now, sodium is positively charged. Now, yes, it's pumping potassium in, but it's pumping a lot more sodium out. If you think about it, if it's pumping out a positive, all these, positively charged ions, the inside of the cell is becoming electronegative. Maintaining that electronegativity is crucial for a neuron. Now, right now, all of those are controlled by these sort of one-way gates. They're continuously pumping sodium out, pumping potassium in. Those are one-way gates. When you want to actually have the cell fire, the neuron fire, you need to open those gates. Because what happens is, when those gates open and all the sodium flows in, we'll talk about that in just a second, but when all that sodium flows in, that's how you get this sudden depolarization burst of electric or charged energy. There are a couple different ways to open that gate. The first, you see ligand gated, is when a neurotransmitter itself comes into the synapse, which we'll discuss in a second, and effectively hits those gates, those closed gates, and opens them wide up. So neurotransmitters, their function there is to basically hit the open signal and unlock those gates and let the sodium flow out. They're also voltage gated, which we'll talk about in a minute, where the actual propagation of a charge is enough to open up the other ones. Mechanically gated are relatively fascinating as well. Mechanically gated respond, and those gates open up to any sort of contact or pressure. If you think about sensory neurons, maybe say on the tips of your fingers, a lot of those are mechanically gated. It is the very act of applying any sort of pressure or change in pressure that causes those gates to open, that causes that sodium to flow in, potassium to flow out, and causes the action potential to fire. And there are also leakage ones, which we could talk about at some point, but they're not particularly relevant to the rest of the discussion. So, all of these different ones operate via different properties. What is the overall what is going on here? So, as mentioned before, all of these neurons are pumping as much sodium outside as they can and pumping a little bit of potassium in. When these gates are open, whether it's be a neurotransmitter, a voltage propagation, mechanical contact, touch, or anything else, those sodium channels open wide up. So the inside of the cell starts out as negative, but all of a sudden all these positive ions start flowing in. Now, not only do they flow in to equalize the charge, but they flow in at such a rate that they actually cause the inside of the cell to become positive. At the same time, potassium is flowing out. Now, it's not enough to equalize the charge, but it's still to a certain extent. So what happens? All the sodium flows in, the inside of the cell becomes positive, all that potassium starts flowing out at the same time that sodium starts getting pumped and starts flowing back out. So it goes from negative to suddenly it hits this huge positive charge, and then it quickly resets, but then becomes hyperpolarized where it's actually overly negative for a second before all those gates slam shut again and the pump starts reestablishing the gradient. So we'll see this in just a second. Now, that period where it has to return to rest and everything starts pumping is called the refractory period. A neuron cannot fire again immediately because it needs to create that gradient. So this is what it looks like. Now see, item 1 there shows this cell at rest. So the neuron is at rest right now. All those little pumps are keeping the sodium out and pulling the potassium in, and it's got a negative charge. You see that negative membrane potential. When the stimulus is applied, whichever it is, those gates flow open. Now watch, that peak starts to rise as all those sodium ions flow in until it reaches an absolute maximum potential right there. Then when it hits that potential, suddenly that potassium starts flowing in the opposite direction, and then suddenly the gate slams shut and all the pumps start going again. You see that hyperpolarization after the voltage falls at 4, between 4 and 5, you see that dip where the inside of the cell actually becomes overly negative. That is that kind of hyperpolarization. It is not until 6, item 6 there, return to rest, that the neuron is capable of firing again. Now that is actually very important when we talk about motor units and impulse, and we talk about, say, muscle twitch and the like. The action potential, the ability of a neuron to fire over and over again, is actually a metric which is studied in several areas of sports science, including things like, anything ranging from maximum muscle contraction to talking about things like even central nervous system fatigue. Some of that has to do with the ability of the muscles to fire. Others have to do with the ability of a neuron to actually continue to fire. Now, this also comes into play when we talk about things like topics like hydration. We talk about things like hyponatremia, sodium depletion. We talk about, again, any sort of electrolyte concentration in the body. Being able to maintain this consistent gradient within the nervous system requires electrolytes. And think about what would happen if neurons do not have enough of these electrolytes to continue maintaining this gradient. So many systems in the body, as we've seen, rely on this sodium-potassium pump. Something as absolutely critical as this, as the simple firing of a nerve, requires large amounts of these ions. Without them, the nerve doesn't fire. Now, let's go back and actually talk about neurons for a second. Now, we said that a neuron, we've got the dendrites flowing in, we've got the soma, and we've got the axon that fires. The axon has those myelin sheets, and you have these nodes of Ranvier in between that are actually exposed segments. Now, first of all, if a nerve wants to fire, we talked about how all this depolarization takes place. Think about, first of all, a relatively short axon that doesn't have the myelin sheath. Now, let's think about what happens. Let's start at the dendrite side. If an action potential fires there, and you get this depolarization, and you get this sort of electrical impulse starting at the dendrite, how is it going to get to the other end of the neuron? Well, what's going to happen is if you think about the sodium channels opening there, and you think about this sudden charge, this change in charge there, what happens is the rest of the neuron, not just at this synapse, which is where the axon connects to the dendrite, but the rest of the neuron has these voltage-gated channels. When these voltage-gated channels sense a sudden change in electronegativity to this, sudden positive charge, they open up and propagate the signal. So what's happening is, if you think about it as almost like a wave, like looking in a stadium. And the wave starts and people go up, and then as the wave travels, you watch it kind of propagate down and around the stadium. That is sort of the way these voltage-gated charges will move the charge. You suddenly get the opening there, you get the impulse, which is where it starts, and you watch the wave propagate around the stadium. Now, two things to remember is, first of all, that wave can take a long time. It is not the quickest process. No matter how quickly an individual sits up and down, they're not going to make the wave go any faster. Now, ordinarily, this is fine. Inside the brain itself, these neurons are so short, these axons are so short, that this slow rate of propagation in a neuron is not a big deal. But if you think about it, think about, again, the very, very long neuron traveling from the base of your spinal column, your sacrum, all the way down to your big toe. If you think about how slowly that signal would likely propagate, it would take a really, really long time for any sort of a nerve impulse to travel down to your big toe. The way these myelin sheaths work is effectively think about the myelin sheath. Now, let's take the stadium analogy. And let's say that rather than a wave traveling down, you had a person every 10 seats apart. As soon as you saw somebody 10 seats down sit up, immediately that signal tells you, sitting 10 seats down, to sit up at the same time. Think about that propagating down the line. Suddenly, rather than waiting for the person next to you to sit up, which would be the way these gated channels would work, you're sitting up the minute somebody 10 seats down does. What's going to happen is that signal is going to travel, that wave is going to travel much, much, much faster. 10 times faster, in fact, since you're doing it 10 seats down. The way these myelin sheaths work is by maintaining an electrical charge, maintaining that sudden positive charge. Every node of Ranvier, when it's activated, when this action potential fires, and at the soma itself, the cell suddenly becomes positively charged. That positive charge travels down the myelin to the next node of Ranvier because the myelin insulates that charge and maintains that sudden positive charge from one node to the next. So rather than it slowly propagating, it's almost instantly propagating, almost at the speed of polarization. Now, this, of course, can't travel the entire length of the cell because when those sodium ions flow in, they can only really cover so much myelin and maintain that charge before that charge dissipates. So those nodes of Ranvier, it would be a little bit like saying, well, what if you were in a stadium so big that you said, okay, I'm going to sit up the minute somebody 10 seats down sits up. Okay, that's fair. Okay, now they say, well, how about 20 seats down? Okay, that's going to be a little tougher. How about 100 and 200 seats down? Can you still do that? You might not even be able to see that person get up. So the distance between the nodes of Ranvier, the thickness of the myelin, the width of the myelin, is more or less dictated by how much sodium can propagate and maintain that charge. So basically, the way these myelin sheaths work is they insulate each portion, and rather than a slow propagation, you get this propagation that goes down between one and the next, and it pops from one node to the next. It happens relatively quickly. They said order of magnitude faster. So that is what allows you to actually activate these very, very long axons relatively quickly. Now, as most of you may be familiar with, there are multiple diseases of myelin out there. Things like multiple sclerosis, Guillain-Barré, a few others, where the myelin sheath, for whatever reason, either starts to break down, isn't correctly formed, becomes damaged due to autoimmune reactions, etc. And a lot of these conditions start to cause various movement dysfunctions, which, if you think about it now, and think about the role of myelin in propagating signals quickly, think about if the episodic signal propagation suddenly becomes attenuated. Or think about if suddenly that these axons, the signal starts propagating more slowly in one area than the next. What would happen if the axon responsible for activating the muscles in your big toe suddenly became significantly slower than the axons responsible for propagating a signal in your ankle? Would you still be able to maintain the same degree of coordination if the signal were even a quarter of a second slower than you were used to? You would suddenly see that you could no longer coordinate those muscles. Some muscles would start firing sooner than others, and suddenly this coordination would come to the point where you could no longer coordinate any motor units whatsoever. That is basically how a lot of these diseases progress. So now looking at the signal between neurons, One of the things we talk about is the synapse. The synapse is basically the region between the axon, which again is the end of the output, and the dendrite, which is the input. Now, keep in mind, certain axons connect directly to other cells and transport the action potential across the synaptic cleft almost directly to another nerve cell soma itself or to the final cell it's targeting. So the receptor does not necessarily have to be a quote-unquote dendrite. However, looking at this diagram right here, we can see exactly what's happening. When this signal propagates down to the end of the axon, the neurotransmitter itself, because between various neurons, we are talking about neurotransmitters. Many of these are, ligand-gated channels. You can see how when the signal propagates down to the end of the axon, there are special voltage-gated calcium channels. Now, there are calcium channels. There are also other neurotransmitter channels. As the signal propagates down, the voltage, these are no longer sodium-potassium pumps that open up at the end of the axon. These are channels that actually then transport receptors, neurotransmitters out. So rather than pumping out sodium, they pump out neurotransmitters at the end of the axon. So this signal transfers all the way down until it gets to the end of the axon, and rather than the axon spitting out sodium, like all the rest of rather than spitting out potassium and bringing in sodium, like all the rest of the neuron itself, it's spitting out neurotransmitters. Those neurotransmitters then travel across this synaptic cleft to a receptor in the dendrite in the next neuron, and then propagate the signal from there. So last topic on the nervous system, peripheral nervous system versus central nervous system. Now, you know how we mentioned that the central nervous system is considered the brain and spinal cord? Much of the CNS is characterized by the fact that the nerves are largely unmyelinated. Try saying that 3 times fast. The absence of myelin, they are much, much shorter. And in fact, there are certain differences in definitions of clusters, basal ganglia versus what they call nuclei, etc. But the main thing to keep in mind is that they are primarily defined by region, by number of connections. If it is not part of the brain and spinal cord, it is considered part of the peripheral nervous system. Now, the peripheral nervous system is further divided into the somatic nervous system and the autonomic. Now, somatic is what we associate with a voluntary movement. When we think of somatic, think about things we want to do and things we actively, consciously process. We talk about touch, any sort of receptor that's responsible for touch, for taste, for movement, muscle, the muscle movement, muscle neurons, motor neurons. Those are all considered part of the somatic peripheral nervous system. The autonomic peripheral nervous system is a lot of what we call the non-conscious function, internal function. This is responsible for a lot of the communications inside the body that we are not completely aware of at any time. If folks are familiar with the sympathetic and parasympathetic nervous system, this is typically considered a subset of the autonomic peripheral nervous system. Sympathetic and parasympathetic we may be familiar with from a lot of discussions on arousal. The sympathetic nervous system considered the, fight, flight, or mating, and the parasympathetic considered rest and digest. Now that's kind of an arbitrary delineation because there are numerous processes in the body and numerous states where they're considered both sympathetic and parasympathetic, but that is primarily what we're talking about now. What that means is that a lot of sympathetic functions have to do with communication between various organs, between the brain and various organs for the secretion of various we call fight or flight related hormones and other system activation. When we talk about the sympathetic nervous system, we talk about fight or flight, we talk about the brain or the muscles experiencing some sort of somatic input from the peripheral nervous system. So let's say I actually touch something sharp or get bitten by something. Well, those sensory neurons, which are, those mechanically gated channels, as part of the peripheral nervous system, send a signal back to my brain, letting me know and letting me process what's actually going on. Now, at the same time as the central nervous system, the brain and spinal cord, are sending the signal to, pull my hand away, at the same time, the autonomic nervous system, kind of without our basic awareness, The sympathetic portion of that nervous system immediately starts sending signals both to certain portions of the brain and then, say, to the adrenal medulla, which is actually part of the, we talk about the adrenal glands. It's sending signals communicating to the brain and from the brain to the adrenal medulla to start releasing certain hormones, such as epinephrine or adrenaline, which will then facilitate my body's ability to react to the stimulus. So whereas the true fight or flight may have a somatic input and a somatic output, it also has an autonomic output. Like I said, in this case, it would likely be a sympathetic reaction. So all of these systems work together, and a lot of them work in parallel. There are certain things, for example, sex and arousal and orgasm, which require both sympathetic and parasympathetic outputs to take place. So there's not always a clear delineation between the two. Now, things get also more complicated when we talk about the digestive system. When we talk about rest and digest, a lot of that used to be considered part of the parasympathetic nervous system, but they're also, at this point, finding that the nervous system around the gut is so complex, and so it operates in many ways independently from a lot of the rest of the system, that people are actually calling it, at this point, some researchers are calling it, they're saying the enteric nervous system is entirely separate. And there are a lot of processes that are actually part of the enteric nervous system that don't fall under the sympathetic or parasympathetic autonomic peripheral nervous system to begin with. You still with me on that? Now, there are a number of organs that secrete various neurotransmitters and components of the nervous system that are not in the brain. So we talk about the brain, we think about, we think about the brain releasing certain hormones and releasing certain neurotransmitters. But again, the adrenal medulla, which is part of the adrenal glands, actually secrete epinephrine. So this is an organ located outside of the brain that is part of the nervous system, effectively, that secretes neurotransmitters. So the nervous system is, of course, much more complex than just the brain and CNS. And other areas, the gut is responsible in many ways for serotonin production. So important to keep in mind, and one of the most important takeaways here, is that the nervous system itself, whereas the brain is sort of it's sort of what's in charge of everything, it is not the only organ that could be considered sort of a primary or even a governor of all hormone release or endocrine release or anything else. So from there, we are going to go directly into endocrinology. We're going to come back and touch on the nervous system later, so specifically looking at, again, motor units and looking at how muscle recruitment works, looking at twitch, maximum voluntary contraction, looking at CNS fatigue and other topics. But endocrine system is going to be next, and we will have some assigned readings on that one. Thank you very much, and that should wrap this one up.